Many students have difficulty understanding how chi-square test is done, so I will use my own example and guide you through the process. Scientific method involves asking scientific questions based on interesting observation. Scientists then formulate a hypothesis to explain why the phenomenon occurred. Once the hypothesis is established, a control test is conducted to prevent unknown variables from affecting the experiment. After the control test, the variable of interest is introduced in the experimental test. When results are gathered, scientists need to determine whether the difference between treated and non-treated groups is significantly different. The chi-square statistical test is used to assess whether the observed difference is truly significant or simply due to random chance. The next slide describes how an experiment is designed based on an interesting observation. One day, when I observe a greater number with bugs on the soil side, I can formulate a null and an alternative hypothesis. After formulating hypotheses, a large pan with two divided areas will be set up for the control test. For the experimental test, brown color is added to simulate the color of soil. An equal number with bugs are placed in the center of the tray, and 2 minutes will be provided so that the organisms can make a choice between two different conditions, brown color and without brown color. When conducting a control test, it is important to confirm that the wood bugs do not prefer one specific side before conducting the experimental test. If they do show a preference during the control test, it indicates the presence of an unknown variable affecting their behavior. Proceeding to the experimental group without identifying and removing that unknown variable may cause the preference to persist, invalidating the result. To remove that unknown variable, students could try a different tray or a different set of wood bugs to make sure a similar number of wood bugs will choose between left or right side in the control test. After you record the results from the control and experimental group, chi-square tests for both groups need to be conducted to see if there is a significant difference in the number of wood bugs from the two different sides. The next slide will show you how to calculate expected total and observed total. When calculating the expected total is based on the null hypothesis being true for both control group and experimental group. As a result, you would expect to see an equal number with bugs choosing the left and right sides. In my example, I use 15 with bugs for replicate 1 and another 15 for replicate 2 in both the control and experimental tests. For both tests, I expect the wood bugs to make equal choices between left and right sides, as they do not show a preference for brown color. Therefore, the expected total for my left side will be 8 plus 7, which is 15, and the same applies to the right side. Now, when calculating the observed total, the number is based on your actual observations. Let's say I observe 6 and 10 with bug preferring the left side. For observed total on the left side, you would add the number with bugs found on the left side in replicate 1 and replicate 2, which would be 16. Once we know the expected total and the observed total, we can conduct a chi-square test to determine if there is a significant difference between the left and right sides. In this case, the chi-square test is performed on the control test. After calculating the chi-square value, you will need to use a degree of freedom and a table of critical values to compare your chi-square value with the critical value. Remember to perform the chi-square test on your experimental test as well. When we finish calculating chi-square value, we need to compare chi-square value to the critical value. To find the critical value, students need to understand how to use a table of critical values. To use this table, we are applying a p-value of 0.05, which means we will accept the result if the probability of result occurring by random chance is 5% or less. In other words, if we perform our control test 100 times, we expect about 5 cases where the result is caused by random chance. These results are considered reliable enough to support our null or alternative hypothesis. You could choose a lower p-value, but it would be much harder to support the hypothesis because you are requiring the result to be caused by an even smaller chance of randomness. Conversely, you could use higher p-value, which makes it easier to support your hypothesis. But some people might question the reliability of your result since you are allowing for a higher probability of random chance in your result. To calculate the degree of freedom, in our experiment, we gave with bugs 2 choices. So 2 choices minus 1 is 1. So we are using 1 degree of freedom. Follow the x-axis at the p-value of 0.05 and the y-axis at 1 degree of freedom. Where x-axis and y-axis intersect is a critical value, which is 3.84. Now, when you compare chi-square value and critical value for the control test, the chi-square value of 0.1333 is less than the critical value of 3.84. Therefore, we support the null hypothesis, which states that with bugs do not show a preference for brown color. Remember, in the control test, we did not provide brown colored side, so we expected to see similar number with bugs on the left and right side. As a result, chi-square test confirms that there is no significant difference between left and right sides, allowing us to proceed to our experimental group. For practice, try calculating the chi-square value for the experimental group and compare it with the critical value. In the end, you should be able to accept alternative hypothesis, which means that with bugs do show a preference for brown color. The grayed out rectangle box contains a solution so that you can double check your work later.